Welcome. Today we're going to explore one of the most fascinating aspects of neurophysiology, how neurons generate and transmit electrical signals. Specifically, we'll examine 3 types of electrical states in neurons: resting potentials, local potentials, and action potentials. By the end of the video, you'll understand how these electrical signals allow your nervous system to communicate at lightning speed. So I provided all my visuals on my iPad. I just drew what I knew. Before we dive into the potentials themselves, we need to understand 2 key concepts: electrical potential and electrical current. An electrical potential is a difference in concentration of charged particles between 2 points. Think of it like water pressure in a pipe. The greater the difference, the stronger the potential. In this example, we see the left chamber has 5 positive cations and the right only has 1 cation. Because there's a huge difference between the 2 chambers, an electrical potential exists that at the dividing wall that's outlined here. If both chambers have equal charges, like 3 cations each, there's no difference and therefore no electrical potential. An electrical current, on the other hand, is the actual flow or movement of charged particles from one point to another. When those cations can move through the wall, we now have a current. Now let's apply these concepts to neurons. In the case of neurons, the 2 points we're comparing are the extracellular fluid, which is labeled here, and the intracellular fluid, which is labeled here. Outside of the neuron. The intracellular fluid is inside the neuron. The dividing wall is the neuron's plasma membrane. The main players in the story are 3 charged particles: sodium ions with a positive charge in the ECF, and then potassium ions also with a positive charge in the ICF, and cellular proteins also in the ICF with a negative charge. Here's what makes this interesting. The ECF has an abundance of sodium ions, making it positively charged. The ICF has an abundance of potassium ions, which are also positive, but it also contains negatively charged proteins that cancel out much of the potassium's positive charge. This makes the ICF negatively charged overall. Here's what makes this interesting. The ECF has an abundance of... Oops, I already read that part. How do these ions maintain their positions? The answer is the sodium-potassium pump. So we can go back, and that will be shown right here. A protein in the plasma membrane that uses ATP energy to actively transport sodium ions out of the cell and potassium ions into the cell. These pumps work continuously like tireless workers, maintaining the electrical state of the neuron. There's one more factor here. The plasma membrane is slightly more permeable to potassium ions, so some naturally diffuse from ICF to ECF, and that contributes to making ECF more positive and the ICF more negative. When we measure the electrical charge of the ICF in a resting neuron, one that's already ready to send a signal but hasn't yet, we get a value of negative 70 millivolts, or negative 70 MV. This is called resting membrane potential. When a neuron maintains that state, sodium outside, potassium inside, ICF at negative 70 millivolts, we say the membrane is in a polarized state. Think of it as a loaded spring, which I have listed here. When a spring is loaded, it's pushed down, then an increased amount of force is going to come and it's going to bounce, as opposed to a relaxed spring, which would not have much, if any, force. Now let's see what happens when a neuron receives a signal. To send electrical signals, neurons need to allow ions to move across the membrane. This is where sodium gates and potassium gates come in, membrane proteins that can open up or close to control ion movement. When a neuron is at rest, these gates are closed. The sodium-potassium pumps keep working, maintaining that negative 70 millivolt resting potential. When a dendrite receives a stimulus, maybe a chemical from another neuron, something interesting happens here. The sodium gates in that region open. Suddenly, sodium ions from the positively charged ECF can diffuse into the negatively charged ICF. And remember, these sodium ions are attracted to the ICF because it has fewer sodium ions and a negative charge. The influx of positive sodium ions disrupts the negative 70 millivolt resting membrane potential, causing it to become more positive. Any shift in the membrane potential is in a positive direction, which is called depolarization. There we go. This disturbance can spread to neighboring regions, causing nearby sodium gates to open as well. This traveling signal is called the local potential, and it moves through the dendrite and into the soma. Local potentials have three important characteristics. First, they are graded. The stronger the stimulus, the more sodium gates open, and the more membrane depolarizes into proportional. It's proportional, like pressing harder on a gas pedal. Second, they're decremental. The signal gets weaker the farther it travels from the site of stimulation, like ripples in a pond, fading as they spread outward. And third, they are reversible. If the stimulation stops, the sodium gates close, the pumps restore the ion balance, and the membrane returns to its resting potential. The spring finally relaxes. Action potentials are the electrical signals that travel down the axon to communicate with target cells, and they're very different from local potentials. First, action potentials follow the all or one none law. Either the local potential reaches threshold and triggers a full strength action potential, or it doesn't reach threshold and nothing happens. There's no in between. It's like a light switch, either on or off. Second, they're non-decremental. Action potentials maintain full strength along the entire length of the axon, whether that's millimeters or over a meter long. Third, they are irreversible. Once the action potential starts, it cannot be stopped, and the spring has been relaxed. Okay, that's where I'm at. Now let's watch an action potential unfold step by step. Oops. Step 1. A local potential reaches the axon hillock and depolarizes it to threshold negative 55 millivolts. Step 2. The abundant sodium gates at the hillock open rapidly. Sodium opens rush into the ICF like a flood breaking through a dam. This causes rapid depolarization. The membrane potential will shoot from negative 55 millivolts, passes through zero, and reaches negative 30 or positive 35 millivolts, the peak of the action potential. Inside of the neuron is actually more positive than the outside. Stage 3. At positive 35 millivolts, the sodium gate closes and the potassium gates open. And now potassium ions in the ICF diffuse out of the ECF. Positive charges are leaving the cell. That will be shown here. This causes rapid repolarization. The membrane potential plummets back into negative values, returning to negative 70 millivolts. Step 4, at negative 70 millivolts, the potassium gates close. Now both gates are closed again, and the sodium-potassium pumps can restore the original ion distribution. How does a signal travel down the length of an axon? It depends on whether or not the axon is myelinated. If unmyelinated axons, the action potential triggers the next region of membrane to undergo its own action potential, which triggers the next region, and so on. The signal travels continuously along the plasma membrane. Think of it like dominoes falling. Each one triggers the next, and while no single domino travels to the end, the signal does. In myelinated axons, it's more complex. Remember, myelin is a lipid protein coating provided by Schwann cells that insulates regions of the axon, but between each Schwann cell is a gap of exposed membrane called a node or a ranvier. When an action potential reaches a myelinated region, the myelin prevents ions from crossing the membrane. Instead, sodium ions that just entered at the node diffuse rapidly downstream through the ICF. Excuse me. They're attracted to the downstream region because it's still at resting potential, fewer sodium ions and negative charge. The signal weakens as it travels through the myelin because the negative proteins attract some of the sodium ions, but just before it weakens too much, the current reaches the next node of ranvier, triggering a fresh action potential. The signal then appears to jump from node to node, which is called saltatory conduction, from the Latin word saltare, meaning to leap. This jumping method is much faster than continuous conduction. Myelinated neurons can conduct signals up to 120 meters per second, and that's close to 270 miles per hour. Now a little recap. The resting membrane potential is the baseline state, a polarized membrane with sodium outside, potassium inside, and an ICF measurement of negative 70 millivolts. The sodium-potassium pumps maintain the state continuously. We also learned that a local potential occurs when dendrites are stimulated, sodium gates open, and the membrane depolarizes. These signals are graded, decremental, and reversible. They weaken as they spread, and they can be stopped. third thing we learned was an action potential occurs when a local potential reaches the axon hillock and crosses the threshold of negative 55 millivolts. These signals follow the all or none law and maintain full strength along the axon and cannot be stopped once started. Together, these electrical events allow neurons to receive information at their dendrites, integrate it in the soma, and transmit rapid, reliable signals down their axons to target cells, whether those are other neurons, muscle cells, or glands. This electrical language of the nervous system, built on the movement of sodium and potassium ions, enables everything from reflexes to thoughts, from sensation to movement. Thanks for watching, and I hope you now have a clear understanding of how neurons generate and transmit electrical signals. Thank you, and have a good night.